Hello friends, today we're going to be talking about DNA. We're going to identify the components of DNA. Know that traits are determined by proteins that are built according to instructions, coded in DNA. We're going to summarize the process of DNA replication and understand that enzymes proofread the newly synthesized DNA, correcting any mistakes. Here's the table of contents. We're going to talk about the history of DNA, its structure, nucleotides, replication, enzymes involved in replication, leading versus lagging strands, semi-conservative replication, and proofreading DNA, as well as talking about an introduction to what's called gel electrophoresis, the importance of gel electrophoresis, principles of, and the setup, step-by-step, data interpretation, and finally, ethical considerations. So let's talk a little bit about the history of DNA. Early scientists thought proteins was the cell's hereditary material because it was more complex than DNA. Remember that we have proteins that are composed of 20 different amino acids in long polypeptide chains held together by peptide bonds. However, some of the first experimentation done by Fred Griffith worked with virulent S and non-virulent R strain pneumonia bacteria. He found that the R strain could become virulent, meaning it is infectious, when it took in DNA from heat-killed S strains. This study suggested, the first study of its kind, suggested that DNA was probably the genetic material. Hershey and Chase, Marcia Chase and Alfred Hershey, they found that chromosomes are made of both DNA and protein. They did some experimentation on bacteriophages, those viruses that kind of look like mechanical little landing craft, by Hershey and Chase, proved that the DNA was the cell's genetic material. This is what those experimentations looked like. So the Griffith transformation on the left, you had a rough strain that was supposedly non-lethal, and the mouse lived. And then you had a smooth strain that was actually virulent, and the mouse ended up dying. So then you heat treated the smooth strain, and the mouse lived. Then you combined the rough and heat-killed, heat-treated smooth strain, and the mouse ended up dying. What that told you was that the DNA was responsible inside that virus that was virulent. Then you had Hershey and Chase did the kind of the same thing. They used radioactivity. They labeled phages with bacteria, and those phages infect bacterial cells. They blended them up, they centrifuged them, they spun it, and they tested for radioactivity, and there was radioactivity in the liquid. Then they turned it green, did the same thing, and then they found that the actual viruses turned green or had that radioactivity inside, and that proved that it was able to carry over that DNA. So then you have DNA structure. DNA structure was proposed by Erwin Chargaff. He showed the amounts of the four bases in DNA: adenine, thymine, cytosine, and guanine. In a body or somatic cell, adenine is about 30.3%. Thymine is also 30.3%. Guanine is 19.5 and cytosine is 19.9. So he came up with what's called the Chargaff's rule. Adenine must pair with thymine and guanine must pair with cytosine. And when we're talking about that, there's an easy auditory device that we use, at least in my class, that apples are in trees, A and T, cars are in garages, C's and G's. Now, the bases were held together by weak hydrogen bonds, and Rosalind Franklin, who some of you have never heard of, took the first X-ray diffraction photographs of DNA crystals. This is what it looks like right here. Notice the double helix. And then Watson and Crick, based upon her work, built the first model of DNA using those X-rays. Now, let's talk a little bit about the structure of DNA. And not all DNA is the same. There is what is called bDNA, aDNA, and zDNA. And if you look at those forms in the drawing left, some of those you've seen look familiar, and then some of those not so much. So let's talk about structure in general. So sides are made up of a pentose sugar, that deoxyribose, bonded to phosphate groups by a specific type of bond called phosphodiester bonds. The center is made of nitrogen bases bonded together by weak hydrogen bonds. Thus, we term it a double helix. Most DNA in the world is bDNA, or a right-hand twist with 10 base pairs and a turn. Left twist DNA is called zDNA, or southpaw DNA, and hotspots can occur, meaning places for mutations, where right and left twisted DNA meet. And so they are producing, or could produce, mutations. And so this is what it looks like, that double helix. The nitrogenous bases are in the middle, and the sugar phosphate groups make up the side or the backbone. And this is a graphical representation of what that might look like. Notice they are what is called anti-parallel. We'll get to that in a moment. But you notice the sugars are facing up on one side. This pentagon is facing up, whereas the pentagon sugar, or the pentose sugar, is facing downward on this side. That's very important, and we'll talk about that in a moment. Now, the actual monomers of DNA are called nucleotides. DNA stands for, once again, deoxyribonucleic acid. They're made of subunits called nucleotides, and these nucleotides have three parts. One is called the phosphate group, and if you look right, you can see that. And you have a pentose sugar, and then you have what's called a nitrogenous base. This is the actual genetic code. Now, some refer to it as molecular bases, but molecular or nitrogenous bases mean the same thing. The double ring, or purines, we call them, are comprised of adenine and guanine. When I say double ring, you see that there are two hexose or pentose sugars that are connected at the nitrogenous base. If it's a single ring, they are called pyrimidines. Thymine, cytosine, uracil all fit that description. You can look right down here at the bottom. Now, the base pairings, purines, only bond with pyrimidines. There are 3 hydrogen bonds. This is very important. 3 hydrogen bonds are required to bond guanine and cytosine, whereas 2 hydrogen bonds are required to bond adenine and thymine, or adenine and uracil. These are what allows for DNA to be copied exactly. Now, notice I've said uracil a couple times. Uracil is only found in RNA. We'll talk about that here probably later on in this unit, but definitely in the next when we talk about protein synthesis, or central dogma. Here is once again that nucleotide structure. You have the side chains made of the sugar phosphate with the nucleotides in between, held together by these hydrogen bonds. If you notice adenine and thymine, right here there are 2, and then guanine and cytosine look like this, and those dotted lines represent the hydrogen bonds. Now, remember we talked about in the cell cycle S phase, or synthesis phase, or where it copies or replicates DNA. Well, there's a certain way that has to be done. So DNA has to be copied before a cell divides. It is copied during S, or synthesis phase of interphase during the cell cycle. New cells will need identical strands. They occur in the nucleus of eukaryotes. And what happens first? You have this replication fork formed by a replication bubble. So at several points along your DNA, you'll have these bubbles that open up, and where replication actually begins is called the replication fork. 2 strands open, forming these forks, these Y-shaped regions. Here's another example right here. New strands grow at these forks. Notice the directionality though. One grows in toward the fork, the other strand grows away from the fork. As 2 DNA strands open up at the origin, these bubbles form. Eukaryotic organisms have many bubbles, whereas prokaryotes, bacterial cells, only have a single bubble. Now, here is the enzymes, a summary of the enzymes responsible for replication, and they are pretty much in order. First one we're going to talk about is helicase. I refer to it as helo just as a nickname, but helicase unwinds and separates the 2 DNA strands at the replication fork, breaking those weak hydrogen bonds. Then you have what are called single-stranded binding proteins that attach to the outside the sugar phosphate backbone, keeping the 2 DNA strands separated and untwisted. Now, I don't know if you've ever done this, but if you twist one end of the rope and the other end of the rope is fixed, guys, it gets tighter. So we need to relieve that tension upstream of replication, and that's where the second enzyme comes into place, called topoisomerase. Topoisomerase is an enzyme that attaches to the 2 forks of the bubble to relieve stress on the DNA as it separates. You don't want it to break. That would be a problem. Then you have RNA primase. RNA primase lays down little segments, little pieces of RNA called primers. These primers present a start recognition for addition of new nucleotides to be laid down. Primase is an enzyme that synthesizes the RNA primer, and then the next enzyme, DNA polymerase, can then attach to and add new nucleotides. So DNA polymerase, we just talked about, can only add nucleotides to the 3' end of the DNA. So when you look at DNA, and I'll show you this better representation here in a minute, it has what's called a 5' to 3' end, and then once again anti-parallel. Those strands lay anti-parallel to one another. The other strand goes from 3' to 5'. So those primes are the number of carbons in the original phosphate group, in the original pentose group at each end of the DNA strand. So new strands, and very important, new strands can only be built in the 5' to 3' direction. This will make more sense when I show it to you graphically here in a moment. Now, the last enzyme is called DNA ligase. Ligate means to connect. And so there are two different strands that are being created, and we'll talk about those in a second. One is called the leading strand. It's called continuous strand or continuous replication. This is constantly moving in toward the fork, while as the discontinuous strand would move away from the fork, laying down these little tiny fragments of newly synthesized DNA. Those fragments are called Okazaki fragments, and by definition, series of short segments on the lagging strand. They can only be joined together by an enzyme called DNA ligase, and I'll show you what that looks like here. Here is helicase moving its way in, starts to separate those hydrogen bonds. The single-stranded binding proteins attach to the outside, and then topoisomerase, represented by this ring, attaches upstream and constantly moves upstream, relieving the stress so it's not twisting and breaking. Here's RNA primase comes in, lays down a little piece of RNA called a primer. Here's the primase laying down that primer, and then DNA polymerase then attaches to that primer and makes a new strand. And then as this helicase continues to move to the right, or in this picture to the right upstream, new DNA is exposed in order to be replicated, and so these are the Okazaki fragments that you keep seeing laid down on this, what's called the lagging strand. On the leading strand, notice it is continuous. There are no breaks. It's because DNA polymerase attached. Notice the new strand is laid down in the 5' to 3' direction, so it is moving toward the replication fork. So how do you remember this? It's a good question. The leading strand is always going to follow helicase. It's always going to be right behind it, whereas the lagging strand is going to be moving away from helicase. There's DNA ligase. It comes in, it removes the RNA primer, and then DNA polymerase is able to fill that gap, making it a continuous strand of newly synthesized DNA. Here's the difference between leading and lagging, a little compare and contrast. The leading strand we refer to as continuous is synthesized by a single strand from the point of origin to the replication fork. And like I said, the original strand in this case, in this representation, goes from 5' to 3', and then the new strand is laid down from 5' at the 3' end of the old strand, 5' back toward the replication fork. The lagging strand is synthesized discontinuously. Notice you have a fragment, you have a fragment, you have a fragment. Well, the first fragment was laid down when helicase was about right here. And as helicase moved this direction, more parent strand DNA was opened up. Therefore, you had to keep making these little segments called Okazaki fragments. Now, we call this mode, this model of replication, the semi-conservative replication or proofreading model. This semi-conservative model of replication idea was presented by Watson and Crick. The two strands of the parental molecule separate, and each acts as a template for the new complementary strand. New DNA of the one parental strand and one new strand of DNA. And that's why we call it semi-conservative. You can see that picture on the left. You have the parent strands, and then add the first generation, it separates, and so you have the purple represented by parent, and then the blue strand represented by the first what is called daughter strand. So how does DNA make sure that it lays down the right nucleotides? Well, we call that proofreading. So DNA polymerase initially makes about 1 in 10,000 base pairing errors, which isn't bad. But these enzymes go back, proofread, and correct these mistakes. Otherwise, you're going to have a change in DNA known as mutation. The new error rate for DNA that has been proofread is 1 in 1 billion base pairs once it goes back and proofreads. Now, why would it need to do that? Well, you can have DNA damage and repair. We talked about that back when we did the cell cycle. We talked about mutagens, chemicals or ultraviolet radiation that damage the body, the actual cells in our body. Cells must be continuously repaired because of DNA damage that we are exposed to. So what happens is they go in and excise, they cut out, refers to over 50 repair enzymes that remove damaged parts of the DNA. DNA polymerase then comes in after it's excised, after it's cut out the damaged pieces. DNA polymerase then goes in and lays down new nucleotides that are normal, and then the DNA ligase puts them together. So this is the actual summary. It goes step by step of what's being laid down. Helicase, topoisomerase, single-stranded binding proteins, primase laying down the primer DNA polymerase, then hooking up to that primase, laying down new nucleotides in 5' to 3' direction, and then ligase connecting all the Okazaki fragments on the discontinuous or lagging strand. Now, why are we talking about DNA replication? Why are we talking about DNA just in general? Well, it's our genetic code, and it is unique for each one of us. And so there is a way to tell your exact quote-unquote fingerprint or genetic or DNA fingerprint. And this is called gel electrophoresis. The definition of gel electrophoresis is a laboratory technique used to separate DNA fragments from other macromolecules, carbohydrates, lipids, proteins, based upon their size and charge. Yes, your DNA has a charge. The purpose of gel electrophoresis is to study, analyze, and study DNA samples, identify genetic variation, differences, and determine fragment sizes. This is what it looks like. The larger fragments obviously are up here at the top. The smaller fragments travel and are down here toward the bottom. The importance of these and applications in research is a fundamental tool in molecular biology. It's used for DNA fingerprinting in forensics labs, genetic profiling, paternity testing, and forensic investigations. Some of you have done Ancestry.com or 23andMe and those kind of things. This is what, or at least part of what they are doing to isolate and determine your genetic makeup. It is essential for studying gene expression. We'll talk about gene expression later, but analyzing protein samples and characterizing mutation. So, some principles of DNA are gel electrophoresis. They are negatively charged due to phosphate groups in their structure. So I just said smaller fragments will migrate or travel faster through the gel. So you see these smaller fragments here at the bottom, and the larger fragments will be at the top. There is a way that this is done. First of all, this medium, this blue brick looking thing, it is agarose gel or seaweed made gel. It's a polysaccharide derived from seaweed used as a medium for this. It forms a porous gelatinous or gel matrix that slows down these fragments when passing electric current through them. So this is the actual apparatus or setup. This is a very simple version. We have machines and computers that do this now. We do PCR and southern blotting and western blotting and all this other stuff. But this is just a general overview of what gel electrophoresis looks like. You have the gel box, you have a power source, you have electrodes attached. Notice when I said DNA is negative, so the negative end is where the DNA fragments are placed. The positive end is where they are going to try to travel to. And there is a liquid inside here that suspends the gel inside the gel box, and the DNA fragments will then travel. The anode or the positive end attaches down here, whereas the cathode attaches up here toward the actual, these are called wells, and that's where the DNA is placed. So this is an example of what it looks like. You have the individual samples here. Usually you have a set of givens or markers on the left side, and you can look at how they match up as you move your way down. And that's how you run, quote unquote, a fingerprint. This is what an actual gel looks like under fluorescent once it's treated with iridium bromide. You put it under UV light and it shows up this way. So the interpretation, identifying DNA bands on a gel. These DNA fragments of different sizes appear as distinct bands. Each band represents a specific fragment size. Smaller DNA bands migrate farther in the gel, larger ones stay pretty close, and those DNA markers that you usually have are samples on the left side as a reference for fragment size determination. These are known fragments of DNA sizes that are loaded alongside the samples. By comparing the migration distances of the DNA bands with the markers, the size of the unknown DNA fragments can be estimated. So once again, this is what this looks like. So I'm taking samples from a crime scene, and I have blood, saliva, any of those kinds of things that contain cells. I can go through and separate all of these out, and I can see which one matches to the ones left behind in the crime scene. And you notice in this case, suspect 2 matches perfectly. Now, comparing DNA samples for genetic variation, we can compare these samples of different individuals or species to identify differences or variations and also their similarities. Gel electrophoresis is employed in DNA fingerprinting in a lot of your forensics labs to create genetic profiles for individuals and for, once again, comparing crime scene DNA and suspect DNA. It's also used to analyze gene expression by separating RNA transcripts, the actual segments of RNA, and we can analyze the proteins that are involved during gene expression. Now, some common issues: smearing, distorted bands, insufficient separation. That just takes some practice in a lab. You can also have problems with the amount of voltage applied or the concentration of the gel, once again fixed with the right lab technique. So, what are some ethical considerations? And we're going to end with this. The ethical considerations of DNA gel electrophoresis and genetic banking and all of these things. The responsible use of DNA analysis, considering privacy. Privacy is a huge deal. 23andMe just got hacked. Their website got hacked about a month ago. So you are releasing millions of people's most personal information, their genetic information, which could be potentially misused. They found all of those people on the dark web that are being sold between $4 and $10 a pop. Now, privacy. Obviously, individuals keep their genetic information private. You don't need your genetics out there just because people can take advantage, like insurance companies and those kind of things, because you can be genetically predisposed to certain diseases that may never show up. Obviously, informed consent is essential before obtaining and using DNA samples for analysis, unless it's a crime scene, unless you are a suspect in a crime. Then usually there's not a consent. They're usually made by court order. Consent should be voluntary, and individuals should have the right to withdraw their consent at any time. Is there potential misuse? Absolutely. Genetic information can reveal sensitive and personal details about individuals. They may face discrimination in employment, insurance coverage, access to certain services. Safeguards should always be in place, and proper handling and disposal of someone's genetic material is essential. Ladies and gentlemen, I hope you enjoyed this lecture, this time we've had together. If you have any questions, make sure you contact your instructor, and we'll see you next time.